This is TWIS. This Week in Science, episode number 889, recorded on Wednesday, August 24th, 2022. How big could lizards get? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with water, drought, and sleep deprivation. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Climate change. It's not a mystery. We know who done it. It was us. How it was done? Relentless, unregulated carbon release. Why it wasn't stopped? Financial interests blocking legislation through lobbying and misinformation campaigns. How bad is it? Pretty darn bad. Thousands of scientific studies have quantified the degree to which we have already altered the climate and committed to changing it for years to come. What are the consequences? Really, really darn bad. Thousands of scientific studies have made increasingly dire predictions based on current observations and deep understanding of the past, including some potential mass extinction events coming our way. What still remains a mystery is how anyone considered a stakeholder in the outcome of the planet's habitable stability would continue to obstruct or deny the changes urgently needed. We have one planet, a good one, the best we've ever seen, and we should do everything in our power to keep it that way as Earthlings, as humans, as the most sentient living creature the universe may ever know. Caretaking our planet is the most sacred duty we have. The only thing possibly more important than maintaining a habitable planet, the only thing we will want, even if we leave this planet or castaways aboard a colony ship bound for nowhere, is enjoying yet another episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science, because that's what we do on This Week in Science. We talk about science that basically happened
In the week.
in the week.
It basically happened.
Right. I mean, it doesn't quite work that way, because the science actually takes usually many years to get to the point of
Wait,
publication.
what?
But these are the
I
stories
thought
that are
all of
published.
these stories that we were covering just only took place within a
It's
week.
hot off the presses.
Presses. That's right. Publication.
Yeah.
Boom. All the things that we love. And we do hope that you will enjoy the show tonight. We have so many fun stories. I have a water world, some gene therapy. Drought is bad. Okay. And artificially detecting things and zapping brains and memory and stuff like that. Justin, what did you bring?
Just good news. Giant lizard edition. Fishy global warming findings. What Americans don't know about Americans. And bipedal old times.
Bipedal. Oh, okay. So, yeah, I'm curious about this one. All right. Blair,
Yeah.
what is the Animal Corner holding in store for us?
Well, hopefully that's not a smear campaign against reptiles, but we'll find out in a minute. In my section, I'm going to talk about another plea for canceling daylight savings time. We'll get to that.
But
then also in the Animal Corner, I have frog brains and bee brains. It's a very brainy episode.
Oh.
Ooh, anybody who is brainy or likes the brains should
Or
love
has
this
one.
show. Or has one. Right? You have a brain? You might like this show too.
Yeah.
Yeah, you.
You, yes, you. I know you
You
have
with the
more than one
squishy
neuron
stuff.
in there.
Come
You
on.
with something bigger than a ganglion. you too can enjoy this show. And this show, as you know, broadcasts live every Wednesday at 8 p.m. Pacific time on YouTube, Facebook, and Twitch. You can find us looking for This Week in Science. You can also find us on Instagram and Twitter and Twitch as TWIS Science, TWISCIENCE. And we are on pretty much every podcast platform out there if you want to watch us or listen to us after the fact. Look for This Week in Science. Our website is twis.org. All right. You want to jump into it? Shall we do it?
Yes. Bring on the science.
Raise the science. All right. This world, our Earth, we think of it as such a wet, watery world. But did you know that really only about 1% of the Earth's mass is water? Isn't that surprising?
So it's because it's all mostly a lot of it anyway seems to be stuck on the surface.
It's stuck on the surface. There is a lot in rocks contained in molecular form. I mean, there's a lot of water on Earth. It's nice and pretty and blue on the surface. We have water vapor in our atmosphere. But when it really comes down to the mass of our planet, we are a rocky planet. We are not considered a water world. But
Lots of magma, I suppose.
yeah, lots of magma. Lots of, other elements that are involved in there. But researchers have been searching for other worlds, exoplanets that are out there that might be covered in water, that might actually be super Earths in size and mass and in terms of how much water they contain. And so some researchers from the Université de Montréal and the Institute for Research on Exoplanets, they've recently announced their discovery of a new exoplanet discovered by TESS, which is TOI 1452b. To be or not to be a water planet? Well, anyway, this water planet is a super Earth. It is larger than our planet, and it's got some really interesting characteristics. One specifically being that it is orbiting one of a two-star system, one sun of a two-star system, one star of a two-star system, and these stars are really, really dim. They have a lot less energy than our sun. So even though this super Earth that was discovered by NASA's Space Telescope, about 100 light years from our own planet, goes around its star about every 11 days. Really, really fast.
Ooh.
Yeah, a year is 11 days
Yeah.
in this water world. Even though it's so close to its planet, or so close to its star that it's able to orbit that fast, it's still within the watery, potentially
The habitable zone because it's not as hot there, right?
vaporized water, they think. I mean, we still have to send James Webb on the case of this exoplanet because they think that this is one of the most likely candidates that they have found for a real water world, not one where the water's frozen, not one where it's water vapor, but actually one where liquid water exists on the surface of the planet, similar to our own.
But it's too
70,
big.
yeah, 70% the size of our planet. So like bigger.
Oh, wait.
70% larger.
70%. Okay.
Larger.
So
plus 70%.
By my back of the envelope napkin math, that means it's going to be 70% harder to get out of bed.
Right, but if you're in water, maybe I've swimming in it already. I don't know.
I need less gravity, not more gravity. I don't need a lot less gravity either, but just a little bit less. Keep looking. Find me a slightly smaller planet, one that makes it so I can get out of bed a little easier. Maybe one that makes it to the point where I can dunk in an NBA regulation.
But then your joints, Justin, your joints and your bone mass. It's a feedback loop. It's the trade-offs are going to end up equaling out in the end anyway.
I don't know if my bone mass is going to be so heavily affected. I spend a lot of time on the couch these days anyway.
It would be a very watery
I'm out
couch.
there running and impacting the impacting the
Yeah.
joints
So
to
these
create that
planets that
feedback
are
loop of getting bone growth, right?
these planets that are suggested to be water worlds or ocean planets, this planet they think has up to 30% water, which is substantially more than we have. And in this super Earth zone, they're thinking that this kind of a planet is a really interesting one to try and find and look at because it's transitional between rocky planets like the inner planets of our solar system and the hot Jupiters, the gassy planets that make up our outer solar system. So this kind of a planet is going to be very, very interesting to take looks at. And, maybe, who knows, find those wonderful hints that could tell us whether or not there is life in, because
Yeah.
we know the universal solvent is water. Things dissolve in water. Life has begun in our planet in water and or in mud anyway. But water might have made it possible in other places like this as well. And it would be very interesting, like you're suggesting, Justin, to discover what kind of life would live and evolve on a planet like this.
Yeah,
Who
and I
knew?
love that we have a new toy
And we have
in the James
And-
Webb, because we've
Yes.
made discoveries like this in a sense, and have been many times over the last decade. We keep finding cool stuff, but now we have a new toy to look at stuff with. Better than
And
we've ever been able to.
this week, James Webb, thank you for that segue. James Webb came up with, they've released some more beautiful pictures of planets in our own solar system. Jupiter has been in James Webb's eye. And in this, we're able to see in infrared using the instruments of the JWST, the aurora of the poles. We're able to see the heat of the great red spot. That is some white hot storminess there, everyone. You see the darkness in other areas, but it's giving us a lot of information about what's happening there. And not only are we looking just at the planet, but they are also taking a look at the ring system and the moons of Jupiter. And we are really being able to see the activity of the planets in our solar system in a brand new light.
Well,
Yeah. Yeah. But not only our own planets, but exoplanets like this new discovery as well are going to be in the JWST's sights. The rings are
What?
Yes. You wouldn't think, right? We don't talk about Jupiter's rings. We talk about Saturn's rings. Jupiter has rings. Uranus has rings. There are these very thin bands of material that are out there, and we just
Dainty, subtle.
they're subtle. Yeah, it's very nice. It's like a very little dusting right there.
JG in the chat room saying it looks blue in infrared. And so the thing I've learned about how they're getting these colorized images is, of course, in the infrared is something beyond the human visual spectrum. So what they do is they take that spectrum of infrared that they're capturing this imaging in, and they break it up into those sections that our cones of our eyes operate in, and they just sort of shift it. They just sort of translate it into human vision spectrum. So it's not
That's
so
nice
much
of
that
them.
they have. It's not so much that they've gone in and colorized things like you would do to a black and white image.
It's like they have different filters to pick things up.
It's that they've actually taken that range of the infrared that they're viewing in and moved it as though it were transposed it as though it were the range of human vision, and that's where those images come from. So,
yeah. And there are people working on these images who are citizen scientists, and it's data. So there's a lot of this that it doesn't come in as pretty pictures. It starts as data, and it has to be turned into the pretty pictures. And there are people around the world, scientists and citizen scientists, who just love to do this work. And a lot of these images were developed by, as the story that I was looking at from NASA's blog, by a citizen scientist, Judy Schmidt of Modesto, California. And she processes these images for NASA and researchers very often.
Cool.
Yeah. Very cool stuff. Justin, what you want
Ooh.
to talk about next?
I've got just good news.
Oh,
The
great.
somewhat reliably positive segment of the show where we look at a subject which might otherwise bring the mood down with a little bit of a positive spin. Just good news, giant lizard edition. The study of climate change-induced mass extinctions in the deep geological past has found that such events aren't all bad. Researchers at the Department of Organismic and Evolutionary Biology at the Museum of Comparative Zoology at Harvard University, exploring the impact of environmental crises on evolution during climatic shifts. They found that caused two of the largest mass extinctions in the history of life at the end of the Permian. The first was 261 million years ago, and then there was another 252 million years ago, which that one alone eliminated 86% of all animal species worldwide. That was a huge extinction event. What they found is these horrific climate-driven mass extinction events, which were global warming events, were just good news for reptiles. Reptiles became the dominant group of vertebrate animals living on land after the 252 million year ago mass extinction. Previous to the Permian global warming extinction events, the vertebrate fauna that dominated was the synapsids, aka ancestors of all modern mammals, which I didn't even know that. The mammal ancestors were the dominant life form before the dinosaurs. I thought we only got our opportunity afterwards, but it turns out we already had our shot and global warming ruined it.
No. People are reptiles too.
After the Permian extinctions, the Triassic period, reptiles evolved at rapid rates, creating an explosion of reptile diversity. This expansion was key to the construction of new ecosystems. These rapid rates of evolution, diversification, according to the new study in Science Advances, began in connection to the steadily increasing global temperatures through a long series of climatic changes that spanned almost 60 million years of the geological record. What's interesting about this is we have sort of classically looked at the expansion of dinosaurs as a result of the mass extinction event, because hey, they opened up all the niches in the food webs and everywhere else, and reptiles rushed in to fill them. What this is saying, though, is that reptiles were already undergoing mass diversification due to the temperature rises ahead of the mass extinction events. So while that did play a catalyst in their development, of course, the diversification that was taking place, this sort of rapid evolution amongst reptiles, started based on the temperature, global temperatures found. So yeah, so just global warming, well, bad for what do they call those the mammals. Might actually not be all bad, and sea life too. Really bad for sea life.
What about reptiles that live in the ocean?
So actually, that's a great question. So they do talk about that because one of the things that they also found was a lot of like smaller dinosaurs or smaller reptiles have the ability to sort of just move, just create new habits, maybe go underground, can have all sorts of things. For some of the large ones, that increased body mass created so much heat that getting into the water became a way to cool off. And so if you're like, people have talked a lot about, maybe those giant sauropods, what classically would look like a brontosaurus kind of a thing. They like maybe they were in the water because of buoyancy to help carry all that weight and that sort of thing. But it could just be also helped in cooling off. And a lot of reptiles started transitioned to being sea reptiles because of the heat. And they also moved really far north. You had dinosaurs up in Arctic regions and stuff, because those were very tropical climates at some point. So yeah, global warming can cause mass extinctions for most species, but actually might be a benefit if you're a reptile.
So you said that these
60 million
reptiles
years.
moved to the poles because
Yeah, some did. Yeah.
But so then isn't that an exposure of a niche and not related specifically to the temperature?
Yes. This is after the fact. So, but
Oh, okay.
what they discovered though was that there was really rapid in some forms of reptiles. I guess the ones that went on to become some of the survivors, actually. Some of the smaller ones had much slower changes. Things that ended up being alligators and turtles, I think, had still maintained a very slow, regular pace of evolution. But many lines of reptiles at the time, because they would look at, looking through the geological record, they would go, how does this keep, how could we keep finding all these new dinosaurs that look related to this one, but it was only like a million years later, and now there's all this different versions of it.
Well, so here's another problem with this though, is that reptiles are a polyphyletic group. So they're not actually all evolutionarily closely related.
No. Correct.
So that's the other kind of weird question I have about this is, do you mean the ancestors of lizards and snakes? Or do you really mean reptiles, meaning also turtles and tortoises and alligators and
So and the group that is tortoises, alligators, and that sort of area were some of the slower evolving ones. If I had read that part.
Yes, but so they were slow. So you're really only talking about the sauropsid ancestor, which is lizards and snakes ancestor, is the one that had this very quick evolution as a result of climate change.
And there's another one that's like extinct.
Are you talking about the tuatara? Because they're endangered. Yes. They are not extinct.
Or tuatara. Yeah. Tuatara was also slow. I think one of the slower.
Right. Ones. So that would make sense because lizards and snakes are sauropsids. They're closely related throughout evolutionary history. But these three other areas of reptiles they're only put in because they're like, oh, they're cold-blooded and they have scales. But really, they're like, they're not, it's not a proper grouping. It's just, it's like we put all the things that look alike in a box sort of thing. But so that makes sense that they would have a different evolutionary
So those ones are slow. But the much larger ancestors of crocodiles, turtles, and dinosaurs could not lose heat as easily and had to quickly change their bodies in order to adapt to the new environmental conditions. So it's just what do you call them? The What was it? What is it? I can't even find it in the story. Not sauropsid, the other one.
Tuataras.
Tuataras were the slow. They maintained the slow evolution thing for some reason.
Okay.
But pretty much everything else was just in relation to the heat had to go through some rapid evolutionary change.
Interesting. Kiki, you're back. We dove real deep into reptile chat while you were missing.
Okay. So you haven't moved ahead.
No, we were about to.
Yeah.
We're going to go on without you.
So do you want to hear my bid as to why daylight savings time needs to end forever? Amen.
Yes, please let us know why you really want to get rid of daylight savings time.
So we have a peer-reviewed publication out of UC Berkeley. This is looking at 3 separate studies looking at the impact of sleep loss on people's willingness to help others. In the first study, scientists placed 24 healthy volunteers in a functional magnetic resonance imager, fMRI, of course, to scan their brains after 8 hours of sleep or after a night of no sleep. They found areas of the brain that form the theory of mind network, which is usually engaged in empathy, or people trying to understand people's wants and needs. Those areas were less active after a sleepless night. That's study number 1. They say, quote, "It's as though these parts of the brain fail to respond when we are trying to interact with other people after not getting enough sleep." Okay, so that's no good. Study number 2, they tracked more than 100 people online over 3 or 4 nights. During that time, they measured the quality of their sleep, how long they slept, how many times they woke up, and then assessed their desire to help others, such as holding an elevator door open, volunteering, or helping an injured stranger on the street. You guessed it. A decrease in the quality of someone's sleep from one night to the next predicted a significant decrease in the desire to help other people. And in the third part of the study, they mined a database of 3 million charitable donations in the United States between 2001 and 2016. The number of donations changed after the transition to daylight savings time. 10% drop in donations immediately following the transition. The same dent in gift giving was not seen in regions that did not change their clocks or when they changed them back. So it was directly related to loss of sleep. So it really looks like lack of sleep impairs basic social conscience, making us less desiring to help other people. So what does this mean? First of all, please stop daylight savings time. It's hurting society. Okay. Second of all, promoting sleep is important. Sleeping in or getting sleep or catching up on sleep does not make you lazy, does not mean you are being inactive. It actually means you are preparing your brain to be an altruistic, helpful member of society. So when sleep is undervalued, that can be a problem societally just for like people on the street, but also, as some of the researchers point out, sleep-deprived doctors, nurses, teachers. This is a problem too, right? If your job is directly related to helping others and being empathetic, and your job is designed for you to miss sleep, that is a problem.
It's a big problem.
Yeah.
Yeah.
So get your sleep. If you had a crummy night's sleep, maybe call out sick. Get your sleep. You need your sleep. That is part of you being a functioning member of society. And also, can we all just stop messing with the clocks, please? It's hurting society.
It is. Messing with the clocks, it reduces, when we do the fall back every year, the spring forward, it's no good. It's no good for anyone. And then we decided, oh no, we're going to go permanent daylight savings. And there has been research from these experiments that we've tried in the past that have shown it's really not a great idea, that it doesn't help people.
Go back to how it was. Why are you doing that?
Yeah, this is a really good argument with scientific evidence for pro-social reasons. Why we need to promote sleep generally.
It would benefit society as a whole.
Just
if
one
everyone
more
got
hour.
some sleep.
Just
Later school starting times. How about later work starting times? How about that?
Well,
why not?
So that's important. I think, okay, how long are kids in school?
6 hours, 7 hours. Yeah.
Those are the same hours people should be working, and maybe a little less, so they can go pick up their kids, drop them off and pick them up.
Well, the system was designed for a stay-at-home parent. That's why.
And how are we still using that system? And I know what they're going to say. Well, we'll just make the kids stay at school for a 9-hour day. No, that's not
but you have to pay. That's the
extra.
You have to pay extra for your seat. It's all part of the system. That's not what this is about, though.
It's all the system, man.
Anyway,
this system is our brains.
So you don't have empathy for all these poor.
Yes, the administrators aren't getting enough sleep and they're making these decisions. You're right.
That's very possible as well. There are probably many work decisions being made as a result of lack of sleep in cultures, work cultures that push people to work and be apart and sleep less, do more. And suddenly we have a lack of empathy in our society. I wonder how that came about. Okay. Yep. Let's talk about something that you're really going to like, with or without sleep, Blair.
Okay.
Researchers at University College London have started a new set up experiments, not working on a lot of kids, but they've published their findings in the journal Brain, in which they are using gene therapy to correct color blindness in children. They
That's
have
awesome.
4 kids with achromatopsia. And achromatopsia is caused by disease-causing variants to one of a few genes. And these particular genes, they create the molecular structure of the color sensing light cells, the cones. So there are fewer cones that can sense light that work within the eye, lots of rods. So the color blindness is complete. Very often these individuals also have nystagmus and other vision problems. They have photosensitivity and other issues. They have used adenovirus gene therapy, which has been proven to work in some other situations for its safety. And so now they're testing it in a limited number of kids to confirm whether or not it works and whether or not it's safe to use. And they had 4 kids between the ages of 10 to 15 years old. So not young, but having grown up with a limited number of cones and a majority of rods within their eyes. They're completely colorblind. They treated one eye out of the 2 eyes on the face, and they compared them also against non-colorblind colorblind individuals who were not treated and normal vision people. And in the process, they didn't show results in all 4 kids, but 2 of the kids ended up after 6 months with brain signals, so showing that there was a signal getting from the cones in the eye to the brain, that there was a changed signal for color receptivity that was, they were unable to distinguish it from normal visioned people.
Wow.
Yeah.
So with this, we may actually be able to salvage some of these humans as productive members of society.
Justin.
Might be able to get that pilot's license after all.
That's right.
There's a couple of jobs that you're not allowed to have. Exactly, Blair. That's not fair. Let's fix that.
Yeah. So this really does confirm the speculation that gene therapy offered to children and adolescents can successfully activate these dormant cone photoreceptors and the pathways they're connected to give light sensations that were never experienced previously.
Cool.
Yeah. So who knows? Hopefully this limited study will grow, and we will see this becoming a treatment moving forward that could lead to full color vision for, or not, or replacement, a significant replacement of the color blindness in children from here forward.
I'm gonna have to look, but I wonder if this is the human trial of the study that we reported on the show years ago in squirrel monkeys. It was gene therapy in the eye. So I'm gonna have to look it up because I feel like it probably is, which I love seeing science progress in real time and move on to those next steps. But I can't wait to hear more about this. It's awesome.
You're like, maybe me, but at least future generations.
Yes. Well, I mean, it's a sex-linked trait, so if I ever have a son, I think he's guaranteed to be colorblind. So this would be helpful.
You're like, so kiddo. Maybe we can
Mom gave you something not so good. I'm sorry.
Sorry about that.
This is my fault.
Yeah. Justin, you got something fishy going on over there?
Very fishy news coming out of the climate arena. International team of researchers has found that approximately 90% of all marine life on Earth will be at risk of extinction in 80 years' time if greenhouse gas emissions are not curbed. In the paper published in the journal Nature Climate Change, the group outlines their study of thousands of marine species and how climate might impact them in the future. Specifically, they looked at 25,000 species, including fish, bacteria, plants, protozoans living in the top 100 meters of the world's ocean. They found that under the worst scenario in which emissions lead to global atmospheric temperatures of 3, which actually isn't the worst scenario for where we're headed. That's right now where we're trending is 3, to 5 degrees Celsius, which is, that's really, really bad. Approximately 90% of all marine life would disappear. Poof, gone, dead. Earth has not seen a die-off as great as these projections since the Great Dying 252 million years ago. Hey, that's when the giant lizards showed up and replaced mammalian ancestors as the dominant vertebrates on the planet from the Just Good News story earlier. Remember that?
It's all coming around.
They also found that if emissions were cut to the extent outlined by the Paris Climate Agreement, which is a thing that we're not currently tracking, keeping global temperature increases to below 2 degrees Celsius, the risk of extinction would be reduced by approximately 98%. That's a big number. So it's all big percentages. It's all big numbers. But it makes it, they're talking about the year 2100, which is the less than 80 years from now. We could lose 90%. Well, it might take a few years longer. They'd all be at risk by. Right? Oh no! If it gets up to the 3 to 5, 90% of all marine life. That to me, I don't know. Now I'm not an expert on food webs and how interconnected the biological life forms are on our planet. But it just seems to me that could be one of those collapsing structures.
Not ideal, for sure.
For sure.
Yeah.
Yeah.
Anyway,
not
really,
good.
really, really bad. We should probably do something about the global warming thing.
Yeah, what should we do? What could we do?
Just stop.
Just
Stop emitting carbons, please. Just knock it off.
Who are you talking to, though? You're not talking to the average listener. You're talking to...
Yeah, I am. I'm talking to everybody. It's everybody. And because you know, there's some people who aren't going to try. You might have to try a little harder because of them. Be the carbon offset you want to see in the world.
Well, on the coattails of your, I guess, the risk, which isn't really great for the fisheries, all the fishes of the planet, researchers realized they really hadn't looked into what happens to plants when they're exposed to drought for long periods of time.
I have a guess.
Yeah. So there's, we have droughts. The western coast of the western states of the United States have droughts a lot. France has been going through a drought, lots of fires. There's droughts all over the place. Some places experience desertification, turn into deserts because they have such extreme droughts. And yet,
hot fudge, no?
Yeah, we haven't really studied specifically what happens to plants during drought and what happens to their ability to store carbon dioxide, their ability to grow, and others. So what do you think happens to plants during drought, Justin? You said you had an idea?
Mm-hmm. So I think they might dry out a lot. And two predictions.
Yeah.
One prediction is I bet you their roots go deeper. And the other prediction is I bet their next generation develops quicker. Because we've seen something like, I remember some story we did like this where when plants in a lab were given a drought-like condition, when they were given water again, that next generation tried to get to, seed again faster than the previous generation. They said, oh, okay, the weather's going to change and it's going to do it forever. Maybe, we don't know, but let's have our next generation try to go to seed quicker so we can take advantage of a shorter wet season. Those are all my predictions.
These are great, great predictions. Well, the researchers reported their initial results from 100 shrubby and grassy sites, and The grassland ecologist Katie Wilkins, who worked on this study, is quoted as saying there was catastrophic loss. That plant productivity in water-starved areas declined by 88%. And Wilkins says, what surprised me was just how dead it was.
Yeah, so it was the much simpler answer of they die.
Yeah, so they in this study they were able to get researchers from all around the world, from Iran, from Germany, from various states in the US, all across the world. They got researchers to use standardized testing methods to limit the amount of precipitation that square meter areas in different research sites would actually receive. And so they were able to block and give and simulate drought conditions for a number of different areas. And so the neat aspect of this is that it's not just one spot, it's worldwide, lots of different biomes where they're looking at stuff like this, like Iran that has struggled with drought, Germany that's a lot wetter. And the difference between the shrubby places and the grassy places, they found that the number one, biodiversity helped to maintain all the plant life and their ability at their productivity, which is also a measure of their ability to store carbon dioxide over the year-long drought-like period. Additionally, shrubby areas did better than grassy areas. The thought is that shrubs, like you were talking about, deep roots, Justin, shrubs have deeper roots than grasses very often. And so those shrubs also might provide some shade, so not as much desiccation occurs to the ground and the roots are able to get a little bit more water. And so they're thinking that areas that have shrubs and grasses may do a lot better because then you have a mix. Germany, those areas, although they were simulating drought conditions, the overall areas had a lot more precipitation. And so generally those areas were a lot more productive. So it also has to do with the kind of general situation that the drought comes into. What kind of historical precipitation and water storage has there been? So there are some very interesting results from this, but the big result is not a question at all. And it's what anybody could say: plants grow less. Plants are less able to store carbon dioxide because they're not growing as much, because a lot of them are dying because they're not getting water.
Yeah.
So.
One interesting thing too that this is making me think of is that California wildflowers tend to be more fire resistant than other plants. They also, exactly, have much deeper roots, and we have whole tree species that are dependent on fire. For they had all the pine tree with the cones, the pine cones, everybody has. Those are seed bombs that go off during a fire. They explode out, and then they are planting themselves in the aftermath of a fire. So for those who are looking at California's drought and fire conditions as going, oh gosh, this is terrible. Yeah, well, we may have gotten there during a wet season, because all of the plants in California
yes and no
really dependent. They show
on fire, but at the same time there are limitations.
Fire, deep roots,
right?
And, they
but that's the problem, is the drought coupled with the fire creates a lot of bad stuff, because they're used to fire, but they're used to fire with seasonal rain. Yeah. And so when you remove the seasonal rain, then the fire goes way out of control.
Sure. But
there's no water to help to germinate the seeds that have just been.
Also that.
Yeah.
So hang on. No. So water. a lot of California poppy is the state flower. That can actually make it through an entire season. Those seeds, those seeds can lay dormant in the soil and then regrow later. So California is used to naturally having drought conditions, is what I'm saying. And it's used to also heavy amounts of rain and fires being able to take place as well. So not everything you're seeing is unusual. What you are seeing is the man-made conditions, the lack of indigenous burning, plus the plus the way that we've developed the
And when it
state,
goes on
and
and on
adding
and
grasses, there
a
didn't
year
used to be grasses in California.
a year drought is fine, perhaps if it's been a number of years that have been
Oh.
rainy. When you get 15 or 16 years of drought, however, suddenly that's a situation that is historically unprecedented, and the plants can't deal with it. So
And you're
Yeah.
diverting water towards the
Yeah.
desert, away from areas where it normally stays. There's a lot going on that we do to kind of tip the scales and make it.
Can we
And
just stop killing the plants, please?
Kiki is screen shared a picture of my garden.
A dead dry
I
garden.
don't have a green thumb. I'll just be honest. I don't know. They need to be watered every day.
Hey everyone, this is This Week in Science. You know what? We like to be watered every week. We like to water your science garden with our science news and discussion. We hope that you find it refreshing and that you will share it with others because, who keeps the fruits of their garden to themselves? You always share those fruits with others. I don't even know what I'm talking about anymore. This is This Week in Science. Thanks so much for listening. Let's talk about COVID right now. Okay.
If we
Who
must.
grows fruit in a garden?
Strawberries?
Yeah.
Garden?
Yeah,
Tomatoes are
It's
fruit.
berry.
Tomatoes are fruits?
No, it's a vegetable. Yes, it's a vegetable.
We're talking about COVID now.
We're talking, yeah, he just doesn't want to talk about COVID. Okay. First off, the presses this week that I thought was a fairly significant study published in Psychiatry Research this week. Researchers analyzed people who had been hospitalized or infected with COVID-19 and the likelihood of developing schizophrenia from either of those situations. And they found an 11% increased risk for the development of schizophrenia with hospitalization from COVID-19, suggesting, and these are their words, that schizophrenia should be assessed as one of the post-COVID-19 sequelae. So,
Okay. So
long COVID?
several-
Hey, that's my schizophrenia. Yes.
Yeah. So 100 issues with that. First of all, schizophrenia is a spectrum of disorders.
But
we
this
understand
is,
this.
and very often we suggest that it's genetically based. So maybe there is a genetic predisposition between people who end up hospitalized with COVID-19, but they found that it was not just infection with COVID-19, but hospitalization from COVID-19. And what this indicates is that the brain infection that COVID-19, SARS-CoV-2 virus, we know can get into the brain and can cause damage. And that damage can be long-term and could potentially lead to the triggering of genetic factors, predispositions that could lead to these situations. Or, or I don't know. They haven't completely, it's not like causal. So in this particular study.
No. Because there's other versions of this that I've heard. COVID is doing this now. Drug use has done this. Having a baby does this. Working certain jobs does this. What my point is, though, it seems to be more of a stress factor that reveals a thing or brings to the surface a thing. That's why, like, it's also a lot of teenagers will get diagnosed for the first time. Well, there's a lot of stress and hormone and things taking place in that time of age where could have just always been there, but it's a manageable or unnoticed situation. So I wouldn't get to the point where I'm thinking that it's causal, like you're saying, but more revealing cases. Situationally
not necessarily,
based on stress.
but with the damage that SARS-CoV-2 can do to the brain, it could be there is like you're saying, pre-existing brain pathology, and it may make individuals more vulnerable to the virus getting into the brain and causing further problems than it would have otherwise, which will then predispose the infection to lead to a severe outcome for COVID-19, and then also to the schizophrenia outcome, which perhaps would not have occurred otherwise.
Yeah. No, right. It wouldn't have
Perhaps.
occurred otherwise. But I mean, it's like, okay, it's like this. Yeah, if you never hit puberty, maybe the schizophrenia didn't also. You know what I'm saying? Like, there's all those factors. But what would
Puberty is
be
something that
interesting?
happens to everybody. COVID-19 is a preventable illness.
What would be more interesting
So,
is that if I'm completely wrong, and
Yeah.
and it turns out there's a way for there's a like this is the thing with cancer. Cancer is caused by some unknown vapor, or
virus,
maybe it's
right? Or whatever. Yeah.
but it can be a virus, can cause cancer. What
Yeah.
if this whole time we've been attacking it as a genetic issue, and it's really a viral infection that causes all cases of schizophrenia? That would be very interesting if that were true.
I don't think
so,
that's
though.
true. That's not true.
I think it's going to be a genetic predisposition, some sort of stochastic evolutionary attempt to have minds that think completely differently. Anyway.
Blair, you had an
Yes.
interesting story about variants.
Yes,
and
our
long
variants
COVID.
of concern. This is a study looking at the alpha, beta, and gamma variants of concern, or VOCs, and how they think they might have originated. And so this is a study by scientists at Emory University and the University of Oxford. And they built a mechanistic theoretical model to study the emergence of variants using existing data and software that they developed. And the model rules out a theory that variants of concern emerge from sustained transmission of acute infections. The idea that basically just like by having so much COVID around and so many people infected by COVID that it was able to mutate just because it was pervasive. Instead, this supports the theory that each variant evolved within a single individual with a chronic infection. So
Wow.
this is the idea that essentially long COVID in a specific individual gave rise to each variant. So what this means is it's important to treat it.
Yeah.
It's important to also, to Kiki's point, not get it to begin with, right? But also, it's important to track those with long COVID to see through genomic surveillance new variants emerge within individuals with long COVID.
There was very early on, very early on. This might have been during the first lockdown, even. We had a story where there was like an 80-year-old woman with leukemia who had presented as infected and was still expelling virus for something like 6 months.
6 months, yeah.
In that condition, why you wouldn't need another host. You've been your virus replicating for 6 months in one host. That's more than enough.
And
then
enough opportunity for
vaccinate that
errors
host. Yeah.
that creates mutations.
Yeah.
You give them medication, you vaccinate the host, but the virus continues doing what it's doing with the immune system, and
this
doesn't
was
defeated.
pre-vaccination too. This lady, but she and she was asymptomatic the entire time too. She didn't get sick. Probably the suppressed immune system didn't create at least from that first generation of COVID, it was a lot of times the immune system overreaction that was taking people to the hospital. Yeah. She didn't experience that.
Yeah, so they said in this article, they said it could be long COVID, but also it could be asymptomatic individuals who are carrying it for months, not knowing it. That's where it could come from, for sure.
Yeah.
But maybe eventually, COVID will turn out like other coronaviruses. We can always hope. A new study published in JAMA this last week, looking at the incubation period of COVID-19 caused by all the different variants of concern, the different SARS-CoV-2 strains, alpha, beta, delta, gamma, Omicron. We're gonna keep going. They found that early on, we knew that the incubation period was much longer. The average pooled incubation period for alpha was 6.57 days, even though for a lot of that, it could have been up to 14 or so days. So the CDC and the World Health Organization, they all recommended at the beginning of the pandemic that if you have been exposed, you should isolate for 2 weeks at least to be sure that you are not going to be a carrier, that you're not asymptomatic, that you're not potentially exposing others because you haven't gotten symptoms yet. And so these researchers looked at these pooled incubation periods from systematic analyses of 141 different articles on these different variants, and they found that Omicron variants are down to 3.42 days. So this is partially because it's a lot more infectious. So a smaller dose infects you faster, infects a larger number of cells faster, or the same dose will get in you a lot faster and make you sick faster. But in that, you're sick faster. And so what this means is that potentially the CDC's new advice of staying home for 5 days is actually potentially more accurate, and that we could, if you've been exposed, stay home, keep monitoring yourself, make sure you're wearing a N95 mask, check and see how it's going before you decide to get out into the public. Yeah. I don't know. Although there are tales and stories these days of the attitudes of the public that COVID is over, and you've got COVID. That's no big deal. Just wear a mask or just come to the party anyway.
Yeah,
it's not a problem.
Well, the other problem is you have to have sick time to be able to not go into work also, which not everyone, at least in the United States, has. So it's that's the other little sticky thing.
Yeah. So the environment is changing for us socially, but my fingers are crossed that even though, Omicron is more transmissible, generally people are having milder infections, which is great. And, the number of incubation days is more similar to other coronaviruses. And it, fingers crossed, it keeps going in this direction, even though there's the possibility of more variants popping up.
I just want my flu shot this winter to be a flu shot / COVID shot. And
that would be great.
Yeah, I'm really hoping everyone gets their stuff together and figures that out in time for.
Well, they've Pfizer and others have put in their bids to have their new variant vaccines ready for fall immunizations. So potentially there will be Omicron vaccines ready for you with your fall flu shot. So maybe it's not in one shot, but you can just make one visit.
Just do both arms.
Super vaccinated. There you go. That's how we're tough. Yeah. It's all good. Yeah. That's enough of COVID. Everybody, it's over. No, it's not over. The show isn't over. We have a lot more to come. This is This Week in Science. This is This Week in Science, and we really appreciate your support. If you are enjoying the show, please head over to TWIS.org and click on our Patreon link to show your love and support for TWIS. Click on that link, and it will take you to our Patreon page where you will be able to choose a level of support to help enable TWIS to keep going. Your support, $10 or more a month. We will thank you by name at the end of the show. And now it is time for the time of This Week in Science that we know and love as Blair's Animal Corner.
He loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
So if you imagine that this is a bee taking a walk, this is a bee on pesticides.
Oh, no.
Okay, and no, so for our audio listeners, I would like you to imagine Blair walking her two little fingers across her hand like a nice little tightrope walker. Do, 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 do. And then those little fingers became a
Right.
drunken, wobbly mess.
Stumbly. Yeah.
Okay.
So this is a study from University of Oxford. And it's looking at essentially the line test that hopefully no one listening has had to do because everyone is responsible and does not drink and drive. But if you were pulled over and you were given the sobriety test, they might have you walk on a line and see if you can do that or if you kind of maybe veer off. That is also a test used by neurologists to diagnose neurological disorders like ataxia, which is where parts of the brain that coordinate movement are impaired. So that's a fun fact. Turns out you could do the same test on bees to see if their brain has been damaged by pesticides.
What?
Yes. So insects have an innate optomotor response, which is how they orient themselves in a straight trajectory when they have something that might steer them off course while they're walking or while they're flying. And so the research was to challenge the optomotor response of walking honeybees to respond accurately and timely to videos of vertical bars that moved from left to right. So the idea was they were kind of walking on a straight line, just like the sobriety test that we were talking about before. And then this kind of like a treadmill almost moves the vertical bars to give the bees an optical illusion to make them think that they have veered off course. So in a healthy optomotor response, the bee's motor system orients them back to the illusion of a straight line. But in these 4 groups of wild-caught forager honeybees, they had some of them drink an unlimited 1.5 molar sucrose solution over 5 days, either pure or contaminated with 50 parts per billion of imidacloprid.
Oh no.
50 parts per billion of sulfoxaflor. The harder it is to say, you know it's a bad chemical, right? And 25 parts per billion imidacloprid and 25 parts per billion sulfoxaflor simultaneously with the imidacloprid. So they took a bunch of pesticides and they fed them to bees. And then they had a control group. That's the easier way to say that, right? So then they had them all walk on these lines. And all bees were less good at responding to the flow of these bars when they were narrow or moving slowly than when we were moving fast or they were wide. But at any width and speed, the bees who had ingested pesticides performed poorly compared to the control. So for an example, they turned quickly in one direction and then didn't respond to the direction in the other way. It's kind of like you veered off course on your sobriety test and fell over. The asymmetry between left and right turns was at least 2.4 times greater for pesticide-exposed bees than for controlled bees. So
That's significant.
they weren't ambi turners for sure. They could only handle one side. So
I'm just gonna keep turning left.
yeah,
Turn in circles.
They also showed that with molecular techniques, that the pesticide-exposed bees had elevated proportions of dead cells in their brain's optic lobes, which is important for processing visual input.
Yeah.
Similarly, the key genes for detoxification were dysregulated after exposure. So they damaged their brains and took away their ability to fix the brain damage, essentially.
And took away the ability to break down the
Yes.
pesticide that was causing the problem. So it's like the pesticide is like, I'm going to break your cleaning equipment. I'm going to stick around here and I'm going to just keep dousing you with dopey stuff and you're not going to do well.
Right. And so all of these specific things they found, though, the dead cells and the detoxification dysregulation, they were variable, highly variable across bees, not in direct correlation with the responses that they saw behaviorally. So that means there's also other things happening in their body that's causing them to act this way. So, looking forward, the researchers say to fully understand the risk of these insecticides, they need to explore whether the effects they observed in walking bees also occur in flying bees. The main concern there is that if they are unable to overcome impairments while flying, obviously that has huge effects on foraging, navigation, and of course, pollination. So, not being able to fly in a straight line would be pretty bad. So, testing the flying is next. But essentially, it's not a surprising finding that ingesting pesticides hurts bees. It hurts us, and we're much bigger. So,
but isn't this kind of the explanation, or part of the explanation behind how the pesticides hurt bees in multiple different ways? So,
right.
We've heard previously for years that pesticides can damage the ability of bees, makes them more sick, right? It makes them unable to get back to the hive. That there are these behavioral effects, and it's like, what is going on? How come they can't get back to the hive? What are they doing? Like, how do they lose their way? Well, they can't go home. They're drunk.
They're drunk. They don't know which way is up. Poor guys.
Yeah.
So
pesticides.
Well, and then is it yes, it's too early. But is it too early to extrapolate then maybe the benefits of having organic produce, or not using insecticides in your own backyard garden, so that you can yourself avoid coming into contact with this sort of thing?
Definitely that. I also saw there were other studies that came out this week about brain problems related to Roundup. I didn't bring that too because I felt it was just too much. But there's pesticides were, I think, by and large approved for use because they were this amazing godsend of, oh my gosh, we can feed everyone. And we're playing catch up now because there's other impacts of these pesticides that were not anticipated at the time.
So the difference, though, that needs to be clarified here is that when you're talking about imidacloprid and other pesticides in that category, it's a pesticide. It's meant to affect insects. It's meant to mimic nicotine. And so mimicking nicotine is something that can definitely affect, pests, insects, mammals, it's all in there, this pathway. So this is something we should be considering closely. However, glyphosate and Roundup, the difference there is that it is an herbicide and it is not meant to affect any molecules that mammals have. So and every study that has maybe not every, I have not done a comprehensive review of the literature, but in speaking over the years with researchers, we have heard that many times that the research on glyphosate and its effects on anything outside of the plant world is greatly overstated. So,
I don't know. I think I've even reported on a few where there was one that I think now that I'm recalling it, it was a little bit of an iffy study because they used Roundup and they found that it killed mosquitoes, but it was also a concentration higher than you would ever use in a garden.
Right.
So
and the study that about glyphosate this week, they're talking about C. elegans experiencing convulsions, but at quantities that are much, much lower than what we would be using in gardens. So the question is, what other things are going? Are there other contributing factors? This needs to be looked at in a larger sense and replicated. We need to look at this stuff very seriously. I'm not on, I love organic. I want organic. I want all that. It's great. We want to live in this world. Can we do herbal and pest control that's natural, where we plant
Yeah.
plants that help to keep certain pests away, and we have plants existing in ecosystems that work together naturally to not to be biodiverse and add to plant productivity. I mean, we have so many issues because we're monoculturing everything. We
Mhm.
have giant fields of lettuce, and that's it, and we have to control them.
Yeah.
Yeah,
No,
our
it's I think
environment is something we've created that we can't manage anymore.
I think my point is still that I think there was a moment where pesticides and herbicides exploded their use, right? And it was definitely something that was like, oh my gosh, this changes everything. And
Mhm.
there was still this overwhelming theory at the time that it's kind of like the dilution theory of like, it's just a little bit. It's by
Yeah.
the time it gets to us, there's so little of it left, and I think that for the animals that live out there, the native plants, but then also ourselves, I think there's a lot more study to be done on the things that we are currently using, and I think that's kind of the thing that I would like to see more of.
Yeah. But we need to really seriously look at stuff like this particular study that you're talking about did,
Yeah.
looking at effects that we have been witnessing for years and years and years. What's it doing? And why is it doing
That
that?
all that said, if you're using Roundup in your own garden, you're just lazy.
Go pull those weeds
Just pull
yourself.
a weed.
All right. Well, let's talk about lazy frogs instead, shall we?
Yeah, let's do
go.
it.
Uh, Talk about camouflage v smarts. So there was a study that came out this week from University of Zurich, along with researchers from China and the Netherlands, investigating how frogs have adapted to selecting pressures on them over their evolutionary history. So there are lots of animals out there that want to eat frogs, many, in fact. And so they studied the evolution of anti-predator adaptation in over 100 species of frogs. What they wanted to see was that there are some species that have an escapist strategy where they use their very big brain to outsmart and outrun a predator. There are others that use camouflage, so they basically just count on the fact that they look like something else and stay nice and still, and that gets them through most of the time. So these two different strategies kind of conflict in their own way, because if you are camouflaged, you cannot be conspicuously colored for mate selection. And so you have to there's a push pull, there's a trade-off, right? Are you going to be brightly colored and really fast and really smart, or are you going to camouflage and just bank on not being eaten as your main draw, I guess?
Or I guess you could be both if you have something in a different visual spectrum than your prey or your predator.
Interesting. I think that happens sometimes. A lot of the time, prey or predator species will evolve the ability to see those those
Yeah.
wavelengths. So, that's its own kind of arms race, right?
Yeah.
So, the findings from this study over 100 different species of frogs was that frogs with few predators may rely on escape strategies to survive. Their relatively large brains allow for a flexible flight response, and their muscular hind legs take them far away very fast. But frogs that have many predators, it's not as helpful to be fast because you're surrounded by bad guys. And so that also takes valuable time away from searching for food if you're constantly running away because there's so many bad guys. So it is more likely that you would have camouflage. Looking at the evolutionary history of these 100 different species, they found, surprisingly, that evolution led in a shift from cognitive predator evasion to camouflage and therefore smaller brains. So these weren't just different strategies that came around at similar times, just like some of them this worked, some of them that worked. No, the suggestion is that camouflage actually isn't an ideal strategy, as it often is talked about when we talk about animals. Camouflage is like the tip top, right? Probably just because we think it's really cool. But it's actually a secondary adaptation for when the energetic costs of being smart and fast are too high. Researchers do want to conduct further research to determine if those links apply to other strategies, like poison glands. Like, where do the poison glands fit in this whole narrative? Or if this similar drift happened in other animal species or groups of species. And so,
I definitely think that this applies to humans 100%, because I've always felt that people who wear camouflage as a fashion choice are less smart.
Interesting. I don't think
now I get it.
to
Now I
worry
get
too
it.
much
It's
about
an
predators,
energy
but
trade-off. You know, if you're not intelligent, just wear camouflage instead.
The brain
There you
is
go.
something that takes the majority of our metabolism. It takes a lot of our metabolism. So having a large brain, especially if you have to keep your body weight low, to jump, to do all sorts of evasion tactics, even though the brain can help you reason those things out potentially at a certain point, or even help with the neural processes for the just instinctual reactions, even at the same time.
Yeah. So
Brains, I can't do it. I just gotta look cute like this leaf. Don't see me.
Researcher Stefan Leupold says, quote, "Our study highlights that research on brain evolution has to consider metabolic costs as much as it focuses on cognitive benefits." So their study further suggests that the changing of habitat of prey can alter energetic investments. So basically, when we're looking at brains, bigger is not always better if the energetic cost can have negative impacts in the long run. So that's an important thing to keep in mind. But I love this idea that we have a kind of a map now of the order of different evolutionary strategies. I think it's fascinating, and I can't wait to hear more about frogs and other species. It's very cool.
More comparisons.
Yeah.
We want them. Justin, you want to start talking about some science?
Yeah, I can do that real quick. Oh, let's go ahead and jump into bipedal old times. Bipedal old times. Bipedal old times. Bipedalism is considered to be an important first step in human evolution. Talking about how these things are prioritized. We see it appear right after chimpanzees in the classic human evolution infographic that goes from chimp to upright caveman to modern man slumped under a table at Starbucks, down on all fours, looking for somewhere to plug in a laptop. But we have known that this unfortunately classic image of chimp to man stuck in most people's minds is false. As chimps and gorillas evolved into knuckle walking only after splitting from a common ancestor with humans, and that human ancestor never went down the knuckle walking path. That's not an ancestor of the current modern humans or ancient humans. A forearm and thigh bone found in the desert of Chad of a 7 million year old human ancestor has been studied. Now, previously, they had found portions of the skull and a jaw and some teeth of this. And from that, they extrapolated that this was an upright, bipedal human ancestor. And just because of how the head would have had to connect, where the connection would have been to the spine, And there was some controversy about it. Some people were like, no, that's not enough information. You can't tell from that. And some people were like, yeah, we're pretty sure. We looked at the data and it seems to fit. Now they have a forearm and a thigh bone. Bunch of measurements. Analysis took place looking at the external morphology and the internal structures using microtomography imaging, biometric measurements, geometric morphometrics, and biomechanical indicators. They compiled all this stuff. The collected data was then compared to current and past ape fossils: chimpanzees, gorillas, orangutans, Miocene apes, members of the human group, Australopithecus, Ardipithecus, ancient Homo, current modern humans. The structure of the thigh bone indicates the creature was usually bipedal, both on the ground and also likely while hanging out in trees. According to the results from the forearm, this bipedalism coexisted in arboreal environments with a form of quadrupedalism, so they could kind of move around on all fours, but it's an arboreal clambering that is enabled by hand grips, firm hand grips, that is different from the strategies used by a modern day chimpanzee. The
Different
conclusion,
from like on the ground quadrupedalism.
or in tree, even the in tree quadrupedalism
It's different.
is different than how a chimpanzee would make its way through a tree. In the hand use. The conclusions of the study, including the identification of habitual bipedalism, reinforce the concept of a very early bipedal locomotion in human history, even while other modes of locomotion were also practiced. I can't say the name of this creature, but I'm going to give it a shot. Sahelanthropus chadensis. You can find this is published in Nature. Study reinforces the idea that bipedalism, that's where we started. It's not a new thing. Yeah.
It's the
So
thing.
we have to get rid of that. We got to get rid of that evolutionary infographic. We got to go find where that thing is and delete it from the internet.
Well, you gotta show the braided stream too. It's just a mess.
Right.
That's the other thing
not
that's missing.
this
You're
direct
right.
path at all.
Because you'll start from this little bipedal tree climbing thing, which is very not even ape-like. Like it's got this very interesting brow that comes out. It's almost like a sun visor of a brow, of bony brow that sticks out. You have to start from that, and then partway through, you have all these others that show up and are like, "Hey, we're all here, part of your history."
Well, and humans aren't the end either, so that's like the other problem is that they're not the last. So then it really just should look like a bunch of hominids at a party, just like.
Yeah. Well, and you're
really
right.
what we need to do is
we
take
hope that we're not the last hominin.
Yeah. Well, I mean, we got to look at
Human
our
hominid.
evolution as not a pyramid leading up to us,
Right.
but basically the other direction where there was an organism and it's branched out and there's now a whole bunch of different organisms still living.
But that braided stream where you would have to have the other lineages of hominins, and then they bump, and then there's this one, and then it bumps over to this one, and then it's a much more confusing map than that little infographic.
Yeah.
But we should at least get rid of the chimp at the beginning of it because it doesn't belong there at all.
Huge problem
there at all.
Yeah.
And then my last story tonight is this is interesting. Pop quiz. Get your number 2 Ticonderosa pencils ready. What percentage of Americans support climate action? What would you guess? What would you percent of Americans who support action on climate? What would you say that is?
70.
Oh, that's a high number.
I would say 60.
60. That's okay. That's both of you say most Americans.
Yeah.
That's because you're kind of in the know. According to a recent study.
Yeah, like you're asking the wrong people.
I know 66 to 80% that they gave a range. How do you do your whole study and then come up with a 14-point range? I don't That
It's because you have like a mostly and then absolutely, and so they have
okay.
it's because it's all qualified, right? So
Yeah.
it's
Well, as
so
long
you
as
have to
it's
kind
anchored a little bit, that's fair.
it's they're doing statistics. There's real reasons behind those percentages for sure.
66 to 80% of Americans support positive climate action. Same poll. What do Americans think the percentage of Americans who support climate action is? That's
30.
37 to 43%.
Yeah, I was gonna say 40.
40.
Yeah.
Yeah. You're right with they should have just asked you.
Yeah,
Because you've got the numbers. I love it.
We are not representative of most
yeah,
Americans.
I've been
I
reading
think.
these climate communication papers for like about 10 years. So it's
This is Greg Sparkman, the paper's first author. He's associate at Princeton at the time, is now assistant professor at Boston College. It's stunning how universal and shared that idea is among every demographic. The researchers, the study did find, as one might suspect, that conservatives underestimated national support for climate policies to the greatest degree, but also the left leaning group believed that a minority of Americans supported climate action as well. The misperception was the norm in every state, across policies, and among every demographic tested, including political affiliation, race, media consumption habits, rural versus suburban. Most Americans think a minority of Americans care about climate change, while the opposite is true.
well, what if you measured the amount of airtime that people get to talk about these things in media?
Yeah.
And talk about the actual positive solutions that are available to us
Right.
and how much opportunity there is. Yeah.
Yeah.
So that's it. So it is a little bit lower in the conservative media digesting diet in terms of perception of how many Americans actually care about this issue. But the fact that it's everywhere tells you exactly that. That's exactly the same conclusion I came to. It is just being underreported.
The loudest voice is not always the most representative.
Well, but
Wise words, Blair.
Yeah, that's I disagree with that. Actually, I think the louder person is usually the one who I will believe. Because the quiet person had the conviction.
If the quiet person had the conviction to yell louder. What?
Yeah, and they would be just shouting it out. But here's the thing. That's
why this study has the result that it has.
Yep.
Some interesting
When
correlations.
social media has the results it does.
If you were in a city or an area where there were visible protests, marches, or actions being taken place, you had a higher evaluation of how many people cared, of what percentage of people cared. If you're out in rural area, maybe where it might be a little bit less. But the fact that most to a super majority of Americans believe we need to take these actions, that's fantastic news. Now we just need to get them to understand that they're not in a minority, that they can talk about it, that their friends and neighbors also think this. And if you have an unpopular or fringe idea, you might be less likely to share it and talk about it and want to gravitate towards doing something. If you realize that, hey, we're all in this together, we're all on the same team, and we all want to do something about this, it should make taking those actions much easier.
Yeah, that's why one of the climate actions that you can take is talk about it more.
Yep.
So that it doesn't feel like a faux pas and it doesn't feel like something that people are alone thinking about, because then climate action is downstream. It's consistent with what everyone wants. You're not swimming upstream trying to get something done by yourself. And it feels more achievable. It feels more hopeful. It feels more possible.
I would venture
more science marches, huh?
I would venture to guess that our audience even beats that 80%. I bet our audience is close to 99 percentile.
Because they'd turn us off if they weren't. Yeah.
Because you wouldn't listen to. I mean, I don't know that how many we've been talking about this for the duration that this show has existed, which is a really long time.
People who listen to TWIS, they're in the know.
We got there ahead of a lot of pro-science people, even in terms of really talking about this issue. And it's still amazing to me that we haven't done more sooner, faster, quicker with the urgency and everything else involved. But now that we know that everybody's on our side, everybody's believing and thinking the things that we're thinking, that's really a fantastic. That's can I change my just good news?
Fix the climate party.
Can I change? This was the just good news segment of the show. Forget the thing with the dinosaurs are going to have a great time once there's mass extinction. This is even better than that good news, is knowing that the American population wants to do something about it. Yeah. Now we just need to convince them that they're not alone.
Mm-hmm. You're not alone.
You're not alone.
No.
Look behind you. Yeah.
You are here with This Week in Science. We're here with you. You're not alone. We're talking about science with you. Thank you for joining us for this episode. If you want a TWIS t-shirt or beach towel or other fun thing, head over to zazzle.com/thisweekinscience. We also have a link on our webpage, twis.org, to our Zazzle store, where there are all sorts of great products that you can peruse and buy and enjoy. Fun times, fun things. Support TWIS, wear cool stuff. Blair made all sorts of lovely things for us. They're wonderful. All right, I have a quick few stories before the end of our show here, talking about some brainy things, because I like brainy stuff. Woo woo! And I did want to go back very quickly to the glyphosate story, and I did say that I thought that there hadn't been a lot of evidence showing glyphosate's negative impacts on mammals, on people. However, there are a number of studies that show that glyphosate may have effects through the GABA system, the GABA A receptors that are very important in our brains in the basal ganglia, specifically for motion, and have been linked to increases in Parkinson's diagnoses. But yes, in the particular study that is published this week in Nature.com, Science Advances. Yeah, Science Advances. It's open access. Scientific Reports. Nature.com's Scientific Reports. You can read their study, and they had concentrations that were 1,000 times diluted from previous findings of neurotoxicity, using over 300-fold less herbicide than the lowest concentration recommended for consumer use. So interesting. We'll see where this goes. That it is potentially impacting through the GABA system is intriguing and concerning. So,
not great.
Yeah, not great at all.
I don't know enough about it to know whether or not it creates a reservoir of use, because
just damages neurons.
When you talk about dilutions in these studies, the critical question is can it concentrate? What does it have a body half-life? Because if you're like, I'm using the safe amount every day, does it bioaccumulate? Yeah. Thank you. That's what I'm looking for. Is there a bioaccumulation factor in there too? Because even if you are using it at a much lower dose than the study, but you've used it 1,000 times, well, there you go.
Yes. All right. So other stories, moving on. The artificial intelligence, speaking of Parkinson's, is being applied to detecting Parkinson's disease. MIT researchers have developed an AI, artificial intelligence device, a neural network that has been trained to detect Parkinson's symptoms from breathing. And it can be used remotely using a radar-like system, which basically has little pings off of your body cavity and can distinguish different breathing patterns and patterns that are indicative of the tremors, stiffness, and slowness that often come along with Parkinson's. And so this is a kind of tool that could be non-invasive. Alternatively, it could be used with a belt, like a heart monitor type belt that is worn around the chest that measures expansion and contraction while people are breathing. But it is highly accurate and could be really useful in discerning the Parkinson's progression and how the disease changes over time, and also be able to remotely send information to a doctor so that that information can be tracked and used to enhance treatments. So good
Cool.
uses of artificial intelligence, not necessarily invading our privacy, but helping us treat our neural disorders. But if you do have a neural disorder like Alzheimer's or something like that, that you're losing your memory, you're not remembering things as well. Researchers have also recently published in Nature Neuroscience their study looking at 150 volunteers between the ages of 65 and 88 who received 18 to 20 minutes of TACS. Do you know what TACS is? TACS is transcranial alternating current stimulation. So electrical stimulation to specific areas of the brain. They were trying to use very specific frequencies of electrical stimulation for a specific time period to specific areas of the brain to see what kind of results they could get. And they determined that individuals were able to recall words from a list of 30 words that was just presented to them minutes before their brain stimulation. It became measurable, these improvements, as soon as 2 days after their brains got zapped. And those improvements lasted for about a month, just after a single brain zap.
So what I'm hearing is that electroshock therapy, while not super useful, was close to being onto something.
It was close to being onto something. And what they're, it's refinement is what's happening here.
More of a blunt object as opposed to
you
very
take a
targeted
sledgehammer
technique. Yeah, exactly.
Yeah. So it's the specificity. It's not the whole brain that's being stimulated. They're really looking at these specific areas, and they're seeing that there are very specific results that are occurring in the specific areas of stimulation. And they're hoping that this work is going to be applicable to Alzheimer's patients and other individuals with memory loss, other impairments, so that we could potentially use these, not invasive in the sense of a needle. You can put a cap on your head and get a little bit of a memory boost, but maybe we can help provide better aging for people as the populations around the globe age more and more. Faster, better, stronger, older people.
You got to keep my memory up so when I'm reporting on the effects of climate change in 2100, I remember these conversations we had today.
That's right. It's incredibly important. You can remember the term transcranial alternating current stimulation. TACS.
I didn't remember that now. I need a zap.
Get it zapped. And finally, this particular study just has a lot of personal interest for me. The study of how we remember where we are in space, how we remember where we are navigating through the world. Our hippocampus is involved in that, along with some other brain regions. But we have place cells, these particular cells that become active in different places to help us create spatial maps of different spaces. So you'll have place cells active that let you map, a room, and then you leave that room, and some of those place cells might be active, but in a new network that help you map a hallway, another room, different spaces. And these neurons, they connect together, these place cells connect together to help us navigate our worlds and know where we are in three-dimensional space. How does this happen? Oh my goodness, this is such a huge question. What about these networks? Well, researchers are really interested in this engram, or this network of neurons that are being activated at the same time. And they're trying to figure out what's going on inside the neurons once you've been plopped into a new location. So imagine that you're in Star Trek and suddenly you have been onto the surface of a new planet. Your brain suddenly has to figure out where it is you have to navigate. You have to create a brand new map of where you are. Okay. And then suddenly Scotty beams you back up to the spaceship, and then your brain's like, oh, okay. But then Scotty says, hey, we got to beam you back down, red shirt. And you're like, oh man.
Oh no.
Oh no.
And if you want to be the red shirt who is not the one who dies, you better remember where that cave with the nasty floor Zack was.
Yeah, so that's where the red shirt named Jackson died. In that cave. It just came down, and then that strange creature like shoved him off the thing. Oh yeah. So if your last name is Jackson, don't go on any away missions into caves.
Don't go into those caves.
You got to use the two-handed punch downward. The only way.
That's the only
That's
way.
the only. I don't know how. You're right though. That's the only place anywhere in any form of real or imagined combat where this put your two hands together and then punch somebody, because that will make all the difference.
Yeah, for sure.
For sure.
But anyway, the transporter. Sorry, back to the mice.
Back to the mice in their transporter. Well, so these mice, you can't really do a transporter. So in this research situation, they put electrodes in the mouse brains while the mice were alive, so they could record while they're active. But then afterwards, then they sliced up the brains, and they had to figure out what was happening inside the cells. While the mice were active, they put them in a virtual reality situation. They basically put them in a 360-degree environment and had them walk on a ball in a loop, so that their environment was being projected on the walls around them, and they had to walk on this ball to be able to navigate wherever they needed to go in a maze that was being projected. It's a pretty interesting behavioral setup, and so this is only possible because researchers from a couple of different labs teamed up to work on this. One, a behavioral lab that works on spatial memory and the behavior in mice, and another lab who works on the cellular signaling and the stuff that's going on at the genetic level. And so they did the behavior stuff. They got the mice to go, oh, I want to go to the reward. Oh, I don't want to go where the no reward is. I want to lick this thing. I don't want to lick this thing. And, to do the behaviors. Anyway, transporter zaps them out. And so then they're looking at the red shirt mouse's brain, and what they were able to determine was that a gene called FOS, there's a family of them, these FOS genes. They're considered immediate early genes. And what happens is these genes get transcribed, and they make a whole bunch of other stuff in the metabolism of the cells, in the transcription process of proteins happen. So they're like the marker of stuff happening. And very often people who study the FOS genes, they use it just as that marker. And they're like, oh, look, you put the thing in there, and then look, the FOS gene's going, that brain cell's active, something's happening. This study is the first time that they have linked causally the fact that the FOS genes are causally responsible for the place cells becoming active and for the actual creation of a sense of space, for the ability of this navigation to be able to take place. And so this is the first time they've been able to actually go, these FOS genes and the proteins they make, this is a little pathway of creating the engram of spatial memory within the brain, which is very exciting. And it's very important. And we can think about this little mouse on a ball having its FOSS expressed in its brain after it's been transported onto its little ball planet, having to navigate around. Humans have FOSS in our neurons as well. And so our place cells very likely have FOSS activity. And so if you are that little red shirt on the planet, you better hope that your FOSS is working.
So that must have pretty good FOSS, because I think I have pretty good spatial memory stuff. I'm curious if there's a sampling of humans that don't have that ability to build that. What happens?
What happens if you don't have FOSS? Right. What happens to those cells?
Are these? Is it just confusing? Like constantly, like walking into a room and you're like, oh, okay, let me look around and see where everything is again.
Or is that just like you keep hitting your shin on the same thing every day in the same place?
I'm never gonna remember this one.
Because that is me, for sure.
Yeah. So these researchers, when they blocked FOSS in some of the mice, they actually kept them from in this study. They knocked FOSS out in a subset of the neurons in the hippocampus, and they saw that the cells had less accurate spatial maps of the environment than nearby neurons with normal FOSS expression. Okay. So the maps in the cells were less stable across days, and they say that means that they were less reliable as memories of the environment. So yeah, a lack of FOSS would make spatial memories harder to form and hold on to. They would slowly they'd go away a lot faster and wouldn't be as accurate. Space blindness. You might not have any FOSS. Yeah. That does it for me. Are we good? Did we do this?
I think we have done it.
Hey, everyone out there. Thank you so much for joining us for another episode of This Week in Science. We are absolutely thrilled that you decided to spend 90 minutes of your day, maybe a little bit longer, with us to enjoy science. I want to give some shout outs to people. Definitely. Thank you to Fada for helping with social media and show descriptions. Getting that done is amazing. Identity 4, you're not recording tonight. I hope that everything is going well. But Identity 4, thank you so much for being there to record our show every week. And Gord and Arnlor, thank you for making sure our chat rooms are doing well. And Rachel, I just want to say thanks for all of your fantastic editing. And oh, that's right, our Patreon sponsors. Couldn't do it without you. Thank you to Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazarb, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Gaurav Sharma, Ragan, Derek Schmidt, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Bryan, Carrington, Matt Beto for Texas, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Sue Doster, Jason Olds, Dave Neighbor, Eric Nappy, Kevin Parachin, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you for all of your support on Patreon. And if you want to support us on Patreon, you can head over to twist.org and click on that Patreon link on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels, as well as twist.org/live.
Hey, do you want to listen to us as a podcast, perhaps while you tend to your organic garden? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
This is busy.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for a bi-decadal newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in that subject line, or your email will be spam filtered into the pattern buffer in our teleporter, and it'll get filtered out by the biofilter. We'll never see it.
Or if you're afraid of transporters travel, you don't have to. You can go into the old Twitter shuttle, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.